have some background already in understanding meiosis, and you have some background in understanding basic genetics according to Gregor Mendel. What I want to do today is talk about something called chromosomal inheritance. That's essentially an idea which tries to connect the two. So I'm just going to review very quickly a little bit about Gregor Mendel, his laws, and what we understand about meiosis, and then try and make a more sophisticated connection between the two. So let's start off first of all about Gregor Mendel. Now, the thing to understand about Gregor Mendel, who did his work in the mid-1800s, was that he had done his work before people really knew what DNA was. They certainly didn't know what chromosomes were, and certainly they had no idea about meiosis. Well, what did Gregor Mendel know? Well, for one thing, he was a very good gardener. He was able to cross plants quite well. He was a very good scientist. He taught biology actually in the monastery, and he made very good observations. You can still go ahead and look up his papers in journals. And the other thing he knew how to do very well was to count. His mathematics were really good. Okay, now here's the first thing he did, which was very, very clever. He went ahead and decided he would use a very simple model, and the model was a pea plant. And the pea plant had 7 good characteristics or phenotypes. For example, size, flower color, seed shape, things like that he could do. Now he was very lucky too in that the characteristics he looked at were one or the other. It was like tall or short for the size. Very easy. It wasn't like eye color where there were all sorts of eye colors. And what he realized right away was the only way he could really scientifically compare one variable at a time was he had to generate true breeding stocks. Now, what are true breeding stocks? Those are plants which, when you use their seeds and you get progeny, who are the offspring, they only look like the parents. So if you have a tall breeding plant, it would only give tall children. And if you had a short breeding seed, it would only give plants that were short. These are true breeding. Now, he didn't know it. We know it today. But the reason why is because they're homozygotes. There's no other possibilities. So here's what he did. He went ahead and crossed true breeding plants which are tall with those which are true breeding as short, and surprised they all came out to be tall. That was very suspicious to him. And then he went ahead and crossed that F1 generation, the first generation of offspring, and again he got mostly tall, but he saw the short again. So this was his conclusion. There is something which controls that phenotype. There's some way that plants inherit from their parents. And he called that thing an allele. Now we know today that an allele is really a gene on a chromosome which goes through meiosis and carries the phenotype. He didn't know that. The second thing he said was that it's always there because he sees the short pop up after a generation skip. So he says it must have been that the tall were dominant and they were obscuring the short. We know, but he didn't know. The reason is that that F1 generation must have been heterozygotes. That is a mix of a dominant and a recessive allele. This is basically what goes on here. He started out with 2 plants in the F1 generation, right? And the F1 generation, here we go, were both heterozygotes. They have the 2 alleles there, right? Big T and little t. Then if you cross them, these are the gametes here that could be generated. And if you recombine those gametes, these are the possibilities, the genotypes that we could see in the plant. And we see that we have 3 which are going to be tall and 1 which are going to be short. So the ratio would be 3 to 1. Okay? So he did true breeders. He saw that no matter what characteristic he did, he always got the same thing. It was always that 3 to 1 ratio. And he thought, well, this can't just be coincidence. It must be because the same thing is controlling the heredity of these traits. Okay, before we go ahead and move on to chromosomal inheritance, let's just quickly review what meiosis is. Meiosis is the process where chromosomes first get doubled and then they get divided, so we create four new haploid cells, that is the sex gametes. And these are the stages which you're probably familiar with already. Last time we went ahead and looked at the pictures just quickly, and you can see that the traits are in prophase here. They're inside the nucleus still. That nucleus starts to break down. Chromosomes can't divide unless they get out of the nucleus. Then they line up here along a midline here, which we call the equator. And over here, centromeres on both sides are building a web-like structure called a spindle, which is going to attach to those chromosomes and eventually separate them. Now, the interesting thing is you can see that they don't just line up randomly. They line up in pairs. So chromosomes which are similar, chromosome 1 and chromosome 2, would line up together, 2 and 2, 3 and 3, and so forth. We call those homologous pairs. Now down here, you can see in Anaphase, that spindle which is attached to the centromere, right, starts to pull those chromosomes apart. And it's pulling them to opposite ends, towards the centrioles, right? As they go further and further apart, we go through telophase, where the cells start to divide. So you can see that right here we're getting a pinching, which we call cleavage. And eventually that will pinch and separate those two cells. And we go through a process called cytokinesis, which means movement of cells. And eventually you get two new daughter cells which form. And you could see also that the nucleus has reformed around the chromosomes. So the result is we end up with two new daughter cells with a complement of chromosomes. Now, the interesting thing, though, is that happens twice in meiosis, so we end up with 4 daughter cells which have half or haploid number of chromosomes. Okay, let's go down here to the next one. So if we toggle on just down to the next slide, let's put all this together in chromosomal inheritance. That is, let's make a relationship between meiosis and between Mendelian genetics. Here we go. Let us suppose that you start out with 2 heterozygous plants, big T, little t. Heterozygous because they have one of each allele, and also big T is dominant over little t, so the phenotype, the appearance, is that of the big T. That is, they are both tall plants. Let's go through the stages. What did Mendel say? Mendel said in his first law, segregation of alleles, that when gametes form, they can only form haploid cells, that is, you can only have one of the chromosomes, and the 2 of them must separate. That is, big T must go one way. So if we have a big T and a little t, and it goes through meiosis, according to his first law, segregation of alleles, you could form a haploid cell which is big T or a haploid cell which is little t. That's his first law, segregation of alleles. You cannot end up with big T, little t together. All right? So that's what happens. So if you have big T and little t, you could end up with a big T or a little t. And for this guy also, a big T and a little t. By the way, we know that mathematics is associative and commutative, so it doesn't really matter where you put the t's in here. Okay, very good. Now we go through fertilization. Fertilization, as we know in an animal life cycle, means the 2 haploid cells, sperm and egg, so this might be sperm and this might be egg, or vice versa, they come together to reform the diploid structure, right? And the diploid structure here would be big T, big T, big T, little t, big T, little t, and little t, little t. Now remember, we'll get to it a little bit later, but his second law, the law of independent assortment, says that all possibilities are equal. Now that doesn't mean that you always end up with the equal number of big and small plants. What it means is that the alleles can combine equally. In other words, if they're half T's, half big T, and half little t, it works based on the mathematics. So, in this case, you would have these combinations just like in a square. But if you count up the phenotypes, you see that the percentages are not equal. So who are the phenotypes here? Well, even though we have 3 different genotypes, genotype big T, genotype little t, and big T, and little, 3 different genotypes, you only have 2 phenotypes, tall or short, right? Who are the tall ones? These guys are tall. These are short. Now remember, Mendel can't see the genotypes. He didn't know about chromosomes, but he can see that these are big plants and these are small, and he sees that the ratio is 3 to 1. And no matter what trait he does, flower color, seed shape, and so on, he always gets 3 to 1 when he crosses the F1 generation with itself. And the reason is, when you cross 2 heterozygotes with simple dominance, and that's what this is, big T wins out over little t, you always get a 3 to 1 ratio. Okay? Let's take a look. Let's try another one. So suppose that I gave you a situation, right? Let's try an example, yeah? Suppose I gave you a situation where Mendel went ahead and crossed 2 plants, and one was big A, little a, right? And the other was, shall we say, little a, little a. This one over here we'd call a heterozygous, and it shows the dominant phenotype. And this is a homozygous recessive. It shows the recessive phenotype. Now, let's take it through meiosis, right? Meiosis says, according to segregation of alleles, right? What alleles could we form? Well, they have to separate. So you could end up with A, big A, or you could end up with little a. Now, how about this guy? Who could you end up with here? Well, there's only one allele you could end up with, little a. Now, I could say little a, but they're all the same, right? So let's make a Punnett square that shows this now. What would be the size of this Punnett square? Well, from the one guy, you have 2 possibilities, but from the other guy, you only have 1 possibility, right? So let's draw this guy. This guy could be big A, little a, right? But from this guy, you could only get little a. Now, I could put another little a down here, but the mathematics just divides by 2, right? Who are the possibilities? Let's fertilize them. Bring the gametes back together. Big A, little a, little a, little a. Okay? What is the ratio? These are big, these are small. It's a 1-to-1 phenotype relationship. Now, here's a really interesting idea that you need to know about that we'll talk about later. We talked about what a true breeding plant is. Here's a really sneaky idea that geneticists use all the time to figure things out. They do what's called a test cross. A test cross. I'm going to write that because that's really important. Test cross. What does a test cross mean? It means whenever you have an unknown, you cross it against a homozygous recessive. Why do you always cross it against that? It's like mixing paint with basic white. Basic white has no effect on it, right? Same thing with a homozygous recessive, little a, little a. No matter what you cross against it, the little a will have no effect, so the results will only be determined by the other guy. That's a really important idea. And we'll see how when we do multi-gene crosses, that's really important. Okay? Let's move on here. So, here we have what we could also do. Let's try this one. This is a homozygous dominant, and we're crossing it with a heterozygous. So what might we get in this situation here? Let's go ahead and cross it. I'm going to go ahead first of all and figure out what the alleles are. Who could this guy give me over here? Well, this guy, which is only big T, could only give me alleles which are big T. Who could this guy give me? Based on the first law, segregation of alleles, I could get a big T or I could get a little t. So let's do the Punnett square, right? What would the size of this square be? Again, a 2 by 1. Big T, little t, right? And this is big T. So who would we get? Big T on fertilization, big T, little t. Kind of interesting. Now what do you see? Well, 2 different genotypes. However, if you look at the phenotypes, the appearance, they're all tall plants. Very difficult to figure that one out, right? So you can't really tell the genotypes all the time by looking at the phenotypes. So the important idea here is the first law, segregation of alleles. Now, Mendel actually got very, very lucky in his experiments because he only looked at plants that only had two different types of alleles, and big T, little t, right? And they were simple dominants. That is, big T wins out over the little t. Now, a lot of people said that, gee, Mendel almost got too lucky. Maybe he faked his data a little bit because there's a lot of situations where this doesn't work. And had he tried something else, we might not have Mendel's laws and understand things the way we do. What are the other possibilities? Well, another possibility is what we call incomplete or partial dominance, right? And that means that you have two alleles, but the heterozygous condition is different. Let's take a look at this. Big R over little w. R is for red, W is for white. R is dominant, W is recessive. R wins out over white. Fair enough. Now, if you take two true breeding flowers, a red flower and a white flower, and cross them, surprise, surprise, you don't get red flowers, you don't get white flowers, you get pink flowers. Why is that? They're all heterozygote offspring, this F1 progeny here, and they do not show the red or white, they show pink, because that is a incomplete or partial dominance. Partial dominance means nobody wins out completely. Red doesn't win out over white. Instead, it's a combination of the two. Now, just so we all understand this, a lot of people think when you mix red and white, you get pink. Yeah, that's true, but that's not what's going on here. It could just as well have been red cross white gives blue flowers, right? And blue could have been the incomplete. So don't go down that road thinking that it's a combination of the two, because it's not. So we have another type of dominance here, right? Now, to make matters even more confusing, right, there is something called co-dominance. And co-dominance means that not just that nobody wins, but that they both win. So let's suppose you have a situation where you have big A and little a, right? Now, normally when you cross them, if it was simple dominance, big A would win. If it was incomplete dominance, big A and little a, the heterozygous, would give you something different. But co-dominance, I'm going to write that word here so you remember this, right? Co-dominance means they both win. And that means that both of the traits show up. Really good example of this is a dog with two color eyes, or a roan horse or cow, or a sheep. Or another example of this would be blood type, where you could have AB blood. That is, they both win. So if I cross A and B, I get type AB. Very simple, right? So there are different types of dominance which would certainly make Mendel's experiments more complex. So let's go ahead and look at the blood example, and we'll finish up on that. Now, the blood example actually throws in another problem that Mendel didn't know, which is that, hey, sometimes you have a lot more than two alleles for the same gene. In fact, most of the time you have a lot of alleles for the same gene, and some of them could be dominant and some of them could be recessive, and it gets very, very confusing. Let's talk about blood type. In blood type, you actually have three different types of alleles. They are called, and they're indicated by a little I, by the way. They are called little I big A, little I big B, right? Oops, sorry about that. And then just the little I itself, or what some people call O. All right? I'm just going to eliminate the I's to make it a little easier here. But what I want you to know is that A and B are both dominant alleles, and O is a recessive allele. And the type of dominance that they have is co-dominance. So let's take a look. Suppose I have an example here. Let's suppose we have a paternity suit. We're trying to find out whose kid is whose, right? And let's suppose you have somebody who has type blood A and O. So they're type A, although they are heterozygous for that. Okay? And let's suppose that they have children with somebody who is type B and B. So let's suppose that they are homozygous, dominant. What are their possible children that they could have, right? Well, they could have type AB. That's interesting. A doesn't win out over B, right? They are codominant. They both win. These would be A, B also. And these would B and O, right? And B and O. And B does win out over O. So what are the possibilities for their children? They could have children who are AB, and they could have children who are type B blood. And what might be the ratios of their children? Ratios of their children would be 1 to 1. Okay? That is a multi-allele codominant problem. Okay? So that should provide enough information then to go ahead and see if we can do simple Mendelian crosses using just a one gene system.